Dear listeners, welcome to the second episode of the podcast about enzymes and molecules. I am Roberta Foresti, Professor of Biochemistry at the University of Paris-Est-Créteil and working as a scientist at the Mondor Institute of Biomedical Research in France. Following the previous episode, where we discussed some aspects of heme oxygenase and its product carbon monoxide, today we will be dissecting the role of another important product of HO's enzymatic activity, the bile pigments biliverdin and bilirubin. Although biliverdin and bilirubin were initially perceived merely as waste products produced by heme oxygenase, we have grown to appreciate that they possess remarkable antioxidant properties, being able to fight the action of peroxy radicals, which are destructive molecules produced in our cells. The relevance of bilirubin as a protective substance has also been confirmed by epidemiological studies in different human populations, showing that the circulation of these metabolites in moderate levels in our body is associated with protection against cardiovascular disease, diabetes, and metabolic dysfunction. One particular scientist significantly contributed to shift the paradigm of biliverdin and bilirubin from potentially toxic substances to crucial protective molecules, and we are really fortunate to have him with us today. So I am delighted to welcome Professor Roland Stocker, PhD, who retired only a few months ago after an outstanding scientific career. Dr. Stocker is truly an international scientist, having trained in Switzerland as a biochemist, in Australia for his PhD, and in the United States for his postdoctoral studies. After a short stint at the University of Bern, Switzerland, he moved permanently to Australia, where for several decades he led research teams at the Heart Research Institute in Sydney, the University of New South Wales, the University of Sydney, the Victor Chang Cardiac Research Institute, and again at the Heart Research Institute. He was elected as a Fellow of the Swiss Academy of Medical Sciences and the Australian Academy of Health and Medical Sciences. He is recognized internationally for his expertise in redox biology, vascular disease, and atherosclerosis, a condition whereby a sticky plaque will form inside our arteries, leading to reduced blood flow in vessels and eventually to heart attacks if left untreated. Today, Dr. Stocker and I will talk about why bilirubin can be toxic, how in normal conditions bilirubin is a powerful protective metabolite, and what was the inspiration to explore the idea that bilirubin possesses antioxidant properties. We will also describe recent interesting efforts to produce bilirubin-based drugs for therapy. We hope you will enjoy our second episode of the podcast and that our conversation with Dr. Stocker will clarify the important role of bilirubin in our health. Hello, Roland.
Hello, Roberta. So happy to be able to join you in this.
Thank you so much.
In mind, yeah.
Yes, how are you?
I'm very well, thank you. I enjoy retirement.
Oh, that's excellent. That's nice to hear. Yes. So let's start with biliverdin and bilirubin. Could you describe what they are and where they come from?
Sure, Roberta. So both biliverdin and bilirubin are colored substances or pigments, as we refer them to. Biliverdin has a green color, bilirubin is golden yellow, and both pigments are generated in our body during the enzymatic degradation of a red pigment called heme through the action of heme oxygenase, which you have already covered in your first podcast. And this is really responsible, the heme is responsible for the red color of blood and red blood cells.
Whenever I teach my students about oxidative stress and I introduce them heme oxygenase, I always tell them that this is the only enzymatic pathway that we can visualize. And we can see that actually heme oxygenase is working when we look at the bruise. because there we can really appreciate the different colors that you were describing just now. Maybe you can elaborate on that.
Well, I can only say they're very colorful in it someday.
So,
but I mean, if I may say it, some additional words. I mean, the spectrum of the colors that can develop actually also indicate or reflect the complexity of heme degradation and then oxidation of the pigments themselves. And, maybe later during this podcast, we'll actually come back to some of these issues. So particularly oxidation of bilirubin is a highly complex process, and it can give rise to many, many substances and eventually very small molecules that no longer have color. And that's then when the bruise, eventually disappears.
I see. Yes. So this evolution of the bruise reflects not only the production of biliverdin, bilirubin, carbon monoxide, and so on, but also the different degradation products that occur after these molecules have
degraded. Yeah.
Yes. Yeah. I never thought about that. That's very interesting. And so, without going forward with this, just another point about this. So this means that in a bruise, in a kind of wound, because in essence, this is what it is. It's a wound that is under the skin. If we think about capillaries breaking and so on, there are obviously continuously the production of these oxidant species, even throughout the production of heme oxygenase and the production of the bilirubin and biliverdin and so on. The wound will continue to produce oxidants that will then interact with biliverdin and bilirubin.
Correct. And of course, one of the oxidants is heme itself.
Yes. So there is this continuous, it's a dynamic, because we like to present everything in schemes, but in reality, it's a dynamic of different processes occurring at the same time. Production of bilirubin from heme, from biliverdin, and interaction, continuous reactions with the oxidants that are produced locally by the stress, basically.
Yeah, correct. And in a situation where you have a bruise, you probably have also a situation where the breakdown of heme is not happening in an extremely controlled manner, like during the phagocytosis of red blood cells, where we know that the iron liberated during the action of heme oxygenase is then stored away in a redox inert manner, so to prevent inadvertent oxidative reactions. But in a bruise, in the extracellular milieu, there is plenty of room for heme-derived iron to participate and engage in oxidative stress-related reactions that contribute to the oxidation, degradation of the bile pigments, biliverdin and bilirubin.
Yes, very interesting. As we mentioned earlier, biliverdin and bilirubin were initially perceived only as waste and potentially toxic products. Could you explain why this is true in certain conditions, and what inspired you to hypothesize that the bile pigments possess antioxidant properties?
Yeah, look, this is a number of questions, so let me just go through them one by one. So let's first talk about waste products, and I would like to distinguish that from the potentially toxic products that you've mentioned. So both biliverdin and bilirubin have been commonly considered waste products, as it was thought that they have no biological functions. This is actually despite the fact that in animals we have known for some time that biliverdin, at least, is an important component. For example, it's important in eggshells of certain birds and frogs. It's present in wings of moths and butterflies, and even the placenta of dogs. And the ability to synthesize pigments such as biliverdin and deposit them in eggshells is actually a process gained during evolution of birds. So this has been well documented. And in the eggshell, the pigmentation is represents an important process with respect to sexual signaling and the physiological and mechanical properties of the pigments. And in animal models of research, biliverdin has been reported to attenuate processes such as graft rejection, for example, organs of the heart. Although it is less clear whether this is due to biliverdin or the result of the conversion of biliverdin to bilirubin. That's actually quite a complex issue. So potential limitation to biological function of biliverdin in humans is the common assumption that except for liver and bile, this pigment does not accumulate to significant concentrations because biliverdin is water-soluble. It can be excreted readily, and it's also, as you know, reduced readily by reductases to bilirubin. So biliverdin still actually has function. So to consider biliverdin as a waste product is actually a little bit more controversial than is the case for bilirubin. So why did we think that bilirubin is a waste product? Well, first of all, as we have already said, bilirubin is formed from biliverdin. But the interesting thing is that the enzymatic reaction actually reflects an anabolic process. So anabolic pathways typically require an input of energy, and they're not found in metabolic degradation pathways or catabolism other than degradation of heme. So heme degradation is, in that sense, is very unique as it has this anabolic step involved. And moreover, as you also know, unlike biliverdin, bilirubin is not soluble in water, and I'm sure we'll come back to that later during this podcast. So because of that, bilirubin then needs to be metabolized further for our body to be able to eliminate it. And that's quite a complicated process simply to make bilirubin water-soluble. And of course, that's also then the problem when we come to the issue of potential therapeutics. So the metabolism of heme and formation of bilirubin really stood out when we started looking at that. And we started wondering whether there is not a situation where this so-called waste product could not have a beneficial function.
Yes.
So in situations where heme degradation to bilirubin is increased and the processes required for bilirubin elimination are decreased, so you have a change at both ends. Under those conditions, very high concentration of the pigment can accumulate in blood. And this can result in jaundice, and it's commonly observed in premature infants because in these premature infants, the glucuronidation pathway required for elimination of bilirubin is undeveloped, and they have a need to convert fetal hemoglobin to adult hemoglobin. So there's a very high turnover or very high rate of degradation of heme. And this then can result in exaggerated jaundice and to a situation where the blood's capacity to bind bilirubin is exhausted, and bilirubin begins to accumulate in the skin, in the eyes, which is quite typical for jaundice, but also the brain. And in the brain, it can cause toxicity by mechanisms that are actually largely still unknown. And it can then be responsible for the clinical symptoms of connectors. But except under such pathological conditions, bilirubin is actually not toxic. So now we come to toxicity. So toxicity data for biliverdin are, to the best of my knowledge, not available. And there is actually no reason to suspect that either pigment of dermal of toxicity for the skin or the eye. And working with these chemicals in the lab, we are there actually very safe to handle, except for the usual precautions for manipulating finely divided solids. So, I don't actually think we can make a general statement that we're dealing with toxic compounds. So under very special situations where there is this high level of jaundice, where the plasma capacity to bind bilirubin becomes exhausted, that's when toxicity can set in. But under most situations, bilirubin and bilirubin are not toxic substances. So I'm talking a lot, but do you also want to tell you a little bit about our hypothesis? So why did we start investigating that? And that goes back to late 1985 to the beginning of 1986. And at that time, I was working in the lab of Professor Bruce Ames. And at that time, he was really interested in metabolic pathways and end products of metabolic pathways in particular. And he studied the action of urate and taurine as end products of metabolic pathways. And he found that both these compounds had antioxidant activity. And at that time, we had a very gifted chemist in the lab, Yorihiro Yamamoto. And when Junkan, as he was called to us, looked at the chemical structure of bilirubin, he said, look, at the central carbon atom C10, that these two hydrogens, that, must be very labile. And we knew already that bilirubin was sensitive to oxidation. So we formed the hypothesis that, like uric acid and taurate, maybe bilirubin also has got antioxidant activity. So we then tested this, and essentially, we just used very simple test tube experiments, and we compared the antioxidant activity of bilirubin towards lipids with that of vitamin E, or more precisely, alpha tocopherol, because at that time, alpha tocopherol was thought to be the most important lipid-soluble antioxidant. And we tested bilirubin against lipid oxidation because bilirubin is known to be lipid-soluble and not water-soluble. And I'm going to just highlight the fact that I think strength of that work was that we used very well defined systems. So we used recrystallized bilirubin, we used purified lipids in combination with a very controlled way to generate a specific type of radical. You've already mentioned them, peroxyl radicals. And we generated these radicals not only in a controlled, but in a measurable manner. And we combined this methodology with a new method that was developed actually by Junkan at the same time in the Bruce Ames lab to measure the primary product of lipid peroxidation, namely lipid hydroperoxides, in a sensitive and quantifiable manner. And it was the combination of these experimental approaches that then allowed us to demonstrate that bilirubin, and to a lesser extent biliverdin, have high radical scavenging activity, and that this activity was pronounced at low but physiologically relevant oxygen concentrations, such that at 2% oxygen, bilirubin started to show better, stronger antioxidant activity than alpha tocopherol, which at that time, as I said, was thought to be the most important lipid-soluble antioxidant. So that's really how we went about it. And I stress the importance of the methodology, because I think when we come to some of the newer strategies used today to develop clinically relevant forms of bilirubin to administer to humans, I think one of the problems that field, in my humble opinion, has is that some of the technology that they use to describe the biological activity of the products that they are generating and proposing to be used instead of bilirubin, actually, they're not well-controlled experiments. And I think that's where the field, has still got quite a significant way to go.
I understand completely your point because, of course, I am an admirer of your papers and your, experimental insights. And I think that the fact that you touch on the methodology is very important because I don't know if you feel the same, that nowadays what you are describing to me is, yes, very, very thorough experimental approaches, and it's biochemistry, being able to look at the products, at quantifying precisely products, at having an excellent control of the experimental conditions, which to me are very important, of course, in all kinds of experiments, but in biochemistry especially, I think. And I feel whenever I see papers published, even though they may be fantastic papers and very interesting from a conceptual point of view, I sometimes feel that there are data and findings that are not really proven from a really methodological point of view. Are we really saying that this substance is doing this? For example, have we actually measured this substance in this organ, in circulation, and so on? And I feel we are missing this in the way science is evolving at the moment. So the fact that your approach was very systematic, you define very well your membranes, the way you generated the peroxyl radicals, the oxygen concentration in your environment, and so on, really shows that the findings you had were really strong, were really robust, and of course then later on all of your findings and all of your data were actually confirmed. So I think your approach was obviously fantastic and very something that we have all to aspire to. So you told us how you were able to demonstrate in your experiments that biliverdin and bilirubin were antioxidants. So you showed in the same experiments basically that bilirubin can incorporate into the cell membranes and protect against the attack of free radicals. And free radicals that are not just produced experimentally, but we also produce when we are exposed to sunlight, for example, or to smoking. So you also demonstrated that bilirubin binds to albumin and not just to cell membranes. And albumin, we know, is a protein that is found is circulating in blood. So bilirubin protects also albumin from the attack by free radicals. So the picture that is emerging here is that bilirubin can protect different cellular structures from the attack of free radicals, and maybe from the damaging effect of other kinds of oxidants. How can we explain this phenomenon? And what do you think are the overall implications for the protection of the organism?
Well, so with regards to the lipids and membranes, I mean, we knew from a long time ago that bilirubin is lipid soluble. And so that's why the most obvious first experiment was to look at an antioxidant activity of bilirubin in a lipid environment. And we happened to use, one of the most common phospholipid species in our experiments, phosphatidylcholine. And, we could readily demonstrate that phosphatidylcholine is protected from radical oxidation by bilirubin. And you mentioned albumin. The reason why we turned to albumin and started considering that an antioxidant activity may extend from lipids to protein is simply because albumin is the transport protein for bilirubin in our circulation. But much more interesting to me was the observation that dichroism studies revealed that when bilirubin binds to albumin, something happens to that central C10 atom that I've already mentioned to you. So in such a way that you don't have the intramolecular hydrogen bonding like you have in bilirubin in an organic solvent, but those ones become disrupted so that you have the two chiral moieties of the bilirubin become twisted. And as a result of that, those two hydrogen atoms at the C10 carbon bridge of bilirubin really stick out and become exposed. And that made us wonder whether that could then enable those hydrogen atoms to engage in radical scavenging. And as a result of that, result in a more specific oxidation by peroxyl radicals than what we have seen in the lipids and the membranes. I should perhaps elaborate here and say that when we looked at oxidation products during oxidation of bilirubin in membranes and lipids, we could not identify readily any oxidation products. What we could observe by eye, going back to the Bruce similarity, is the disappearance of color. When we then did the experiment with albumin, it was a complete albumin-bound bilirubin. The picture was completely different. Our eyes told us immediately that yellow turned into green before it became colorless. So we then showed with analytical techniques that indeed when the bilirubin is bound to albumin, oxidation by peroxyl radicals is much more specific than for oxidation of bilirubin in the lipid environment. And biliverdin is formed almost stoichiometrically at the beginning of the reaction before biliverdin and then the remaining bilirubin are oxidized to further secondary oxidation products. And so again, we not only showed that the bilirubin could protect the protein from oxidation, we used quantitative amino acid analysis to demonstrate that. So again, using very quantitative methodology to prove that case. But we actually also found that other ligands of albumin, including fatty acids, were protected by albumin-bound bilirubin from becoming oxidized. And we used linoleic acid as an example. So linoleic acid, dihydrolinoleic acid, is transported also bound to albumin. And yes, when bilirubin is present on the albumin molecules, those linoleic acid molecules are protected from oxidation. And we could observe that this happened under physiologically relevant concentrations of bilirubin, at least physiologically relevant for humans in that concentration range. And, we then extended the studies to other structures in terms of testing the possibility that bilirubin may protect other structures. And we spent several years investigating how bilirubin protects lipoprotein lipids, and in fact lipoprotein proteins from oxidation. This was done in the context of the oxidation theory of atherosclerosis, according to which lipoprotein oxidation is an initial event in atherosclerosis. So we were very interested to see whether bilirubin can protect lipoprotein associated lipids, particularly LDL associated lipids from peroxidation. We could demonstrate that this was indeed the case. And then this also led us to very substantive investigations in the interaction of bilirubin with alpha tocopherol, which happens to be transported in lipoproteins in our body. And that led to some rather surprising and very interesting findings. So, and then other laboratories have extended the antioxidant protective activity of bilirubin to other structures, including DNA, for example, and so on. Proteins other than albumin are protected from oxidation when they bind bilirubin, and so on. So yes, in very general terms, we believe that bilirubin protects both lipids and proteins from a radical-induced oxidation.
Just to clarify with you, bilirubin has to be bound to these structures in order to provide this antioxidant activity, or you think the vicinity of bilirubin to this structure is already enough to exert?
So that's a really, really good question. And again, I think that's related to, potential therapeutics that are being developed. So in the case of albumin-bound bilirubin, that's the situation we studied most extensively. Obviously, the concentration of unbound bilirubin in a solution where you have the protein and the pigment is very, very low. So I think it's very, very simple to assume that it's the bound bilirubin that provides the protection, not the one that dissociates from the protein molecule. Once you move to other proteins, obviously association and dissociation equilibrium is different. Whereas in the case of albumin, it's very much favoring association. With other proteins, the equilibrium is a little bit more is shifted to some extent towards dissociation, but still the majority of the pigment will always be protein bound. So we assume for other proteins, it's also the case that it's the bound pigment that provides the antioxidant protection. Is this just, a localized antioxidant action? Well, we've evidence to indicate that while we do have localized antioxidant activity, the antioxidant activity may not be limited to a local environment. And the best example that I can probably give you is when we did experiments with lipoproteins in conjunction with albumin-bound bilirubin, we could still demonstrate that albumin-bound bilirubin could chemically interact with vitamin E located within the lipoprotein particle. So with other words, there was the ability of bilirubin to provide hydrogen atoms to an alpha tocopheroxyl radical to restore alpha tocopherol and thereby provide a bridge between protein and lipid phase. Now, is there an intermediary aqueous form of bilirubin which is involved? We can't exclude that totally, but I think it's very unlikely. So in summary, I think what's happening is that bilirubin has got two main functions. It has a function as a protective agent for protein when it's bound to the proteins, and it has got a protective function from lipids when it is incorporated within the lipids. And then there is this kind of intermediary scenario also where an albumin-bound bilirubin, we have not tested that for other proteins, but an albumin-bound bilirubin can interact with the lipid phase. Let's leave it at that. But we've no good evidence that water-soluble forms of bilirubin in biological systems provide. So let me rephrase it. We have no evidence that hydrated bilirubin has antioxidant activity to a biological meaning. So we think it's either lipid-bound or protein-bound bilirubin, which has got the protective activity.
So this leads me to the epidemiological studies. We are going from the very, very fine details of the antioxidant activity of bilirubin to the epidemiological studies showing that in healthy populations, a higher level of bilirubin in blood, which is still in the normal range, are associated with a reduced risk of cardiovascular morbidity and mortality. And there are also published studies demonstrating that subjects with diabetes, but that also suffer at the same time from another kind of condition, it's a hereditary disorder called Gilbert syndrome, they exhibit less negative complications from diabetes compared to patients that have diabetes but don't have Gilbert syndrome. So, based on all that you told us today, how do you think your discovery of bilirubin as being a powerful antioxidant fit with these findings in humans?
Well, I think they fit very nicely with this. So you're correct about the epidemiological studies. So higher levels of bilirubin are associated with a decreased cardiovascular mortality and morbidity. And you mentioned Gilbert syndrome. I pronounce it Gilbert syndrome, which is a hereditary condition where the conjugation, the glucuronidation required for the removal of bilirubin from our body is decreased, and that then slightly elevates bilirubin conditions. I should perhaps also say, I'm probably allowed to do that, one of the key interests that Bruce Ames had in bilirubin is the fact that he's actually a Gilbert. He's got Gilbert. So elevated bilirubin, and you also, I think I must be well protected from oxidative damage. But anyway, that's just yeah. I just want to also say that it's the mildly elevated bilirubin concentration as a consequence of Gilbert. It's not really something that the subjects with Gilbert syndrome suffers from. In fact, most Gilbert patients with or subjects with Gilbert are not aware that they have Gilbert. It's a harmless condition, with other words. And you also stated correct that people with Gilbert syndrome and diabetes have got a lower incidence of cardiovascular complications. And in fact, while we have done very little epidemiological studies ourselves, Lab actually has done one piece of epidemiology in diabetic subjects, and we did not look at Gilbert syndrome. We just looked at the general population. There was a rather large study involving nearly 10,000 subjects, and we showed that in people without Gilbert, there was also an association between increased bilirubin concentrations and limb amputation. So the higher the bilirubin concentration, the lower the risk for these diabetic subjects to have to undergo amputation. And as you know, that's one of the peripheral artery diseases, one of the complicating factors in people with type 2 diabetes. So really this suggests that elevated circulating concentration of bilirubin can protect against peripheral artery disease. And of course, our finding fits well, or our basic science findings fit well with that, because lipid and protein oxidation are thought to be contributing factors to atherosclerosis. I've already alluded to this. Atherosclerosis itself is the major contributor to cardiovascular disease, and it's also a very significant contributor to peripheral vascular disease or peripheral artery disease. So I think that's all very congruent. But we probably should also say that bilirubin, in addition to its antioxidant activity, has also anti-inflammatory activities. And we know that inflammation also contributes to atherosclerosis. So we need to also consider that an anti-inflammatory activity of bilirubin may be relevant when it comes to these epidemiological studies. And to me, actually, an interesting question still to be addressed is whether, and if so, to what extent, the antioxidant activities of bilirubin contribute to its anti-inflammatory activities in vivo. That's something that I think people have not really tried to investigate, but I think it would be interesting to find out.
Yes, you're giving me ideas, and I'm not retired yet, so I can still do some experiments in the lab. Actually, you preceded my question because, we are really focused on the antioxidant properties of these bile pigments. And we know even from some experimental models that there are some anti-inflammatory activities of bilirubin, but really we haven't investigated this very, very well. So, and we cannot exclude, of course, that there could be many different actions of the bile pigment that help us to improve and to be protected by these molecules. So we were saying that bilirubin is beneficial against atherosclerosis and cardiovascular disease. And recently you published a very, very elegant work where you described how bilirubin, you went into the mechanisms. You were able to demonstrate that bilirubin is really protective against atherosclerosis using these very elegant models. So I think you really advanced again the field, shedding some light on the protective properties of bilirubin in the cardiovascular system. Can you describe what were your findings and how you were able to demonstrate these findings in your latest work?
Yeah, sure. Look, I need to first just briefly talk about atherosclerosis. You have mentioned it in the introduction, but atherosclerosis contributes to cardiovascular disease outcomes principally in two ways. The first is the way that you mentioned already, and that is that as the size of the plaque, atherosclerotic plaque increases, the lumen within the artery can narrow. And it can narrow to an extent that results in insufficient blood supply to the downstream organ, such as the heart. But importantly, this type of atherosclerosis disease can be readily detected and treated. So there's a second type of atherosclerotic plaques, and they are thought to contribute to probably about half of the events, cardiovascular disease events. And that type of atherosclerosis refers to an unstable and high-risk plaques. And these are characterized by the fact that they can rupture and lead to a very acute cardiovascular event. In the first mode of atherosclerosis, that's a gradual process that people become aware of. Whereas this one here, the plaque rupture, is a very abrupt event and it can lead to death. And unfortunately, at the moment, unstable plaques cannot be identified readily. And there's also no generally available treatment for this type of atherosclerotic disease. So that's where atherosclerosis research really focuses on. So how did we go about to address this question? So we focused on this second type of atherosclerosis, namely the unstable plaque. And in 2013, an Australian group down in Melbourne, they published a really interesting methodology paper. See, again, it goes back to methodologies. And they published a mouse model of unstable plaque. If you want, I can elaborate how the model works, but maybe for the sake of time, I won't. And so we then asked the question with this mouse model, whether bilirubin could affect plaque stability. So that's a different question to ask, can bilirubin affect atherosclerosis? So we generated a mouse model of bilirubin deficiency by deleting the gene encoding biliverdin reductase, which is responsible for the conversion of biliverdin into bilirubin. And this biliverdin reductase knockout mice, they have essentially no bilirubin in their body. And then we crossed these mice with the apolipoprotein E gene knockout mice that is required for the mouse model of unstable plaque. And we look at the development of unstable plaque in bilirubin deficient versus bilirubin proficient animals. And we used very well and established parameters, including histological parameters, etc. And this experimental approach showed that animals that were deficient in bilirubin, they had their unstable plaques became even more unstable. It had no effect on atherosclerosis at sites where stable plaques are being developed. So the effect of bilirubin deficiency was specific, selective for unstable plaque. And it really then showed that in this model, bilirubin has an in vivo protective activity. And we could start uncovering the mode of action. And again, what we observed is this was associated with an increase in lipid oxidation, an increase in protein oxidation, but also with an increase in inflammation. And one enzyme, a pro-inflammatory enzyme that we are particularly interested in is myeloperoxidase, and we could show that bilirubin deficient animals in the unstable plaque had an elevated activity of myeloperoxidase. And this is interesting because in separate studies, we show that plaque activity of myeloperoxidase is a cause of plaque destabilization. And in fact, we're just about to submit the manuscript where we show that plaque MDR activity is also is a predictor of plaque rupture and atherosclerosis. So we believe that in this model, bilirubin then, the implementation is that bilirubin increases should then increase plaque stability. And of course, that is the next step that needs to be shown. And I'm very happy to say that one of my former postdoctoral scientists, Dr. Wei Cheng, is now an independent investigator back in China, and that is going to be the research area that his laboratory will be focusing on.
All this very clear explanation is stimulating another couple of questions that were not planned, but I think I have to ask them. The first is that in this animal model where you prevent bilirubin formation because you are deleting biliverdin reductase, have you seen an accumulation of biliverdin?
Yes.
Yes. So
biliverdin does accumulate, but when it accumulates, its concentrations are still lower than that of bilirubin. And we think that, again, has got to do with the fact that elimination of biliverdin through water-soluble processes is faster for biliverdin than it is for bilirubin. So maybe putting it in different words, bilirubin hangs around as a molecule longer than does biliverdin. And if you want it to be active in a process, that is beneficial if you hang around rather than if you're being secreted very rapidly. The chance that you can provide protection is decreased.
Exactly, because this was going to be my second question or my second comment.
Can I just sorry, Roberto, to interrupt, but initially, very early on, I said, in terms of function of biliverdin in humans, we don't have good evidence for that scenario. And this has been driven primarily by the concept that biliverdin does not accumulate. But with these mouse experiments, obviously we looked for biliverdin, and we started looking for biliverdin not just in atherosclerotic plaque, but also in other tissue. And one of the things that we observed is that, contrary to the common belief that biliverdin does not accumulate in most tissues, we could actually detect biliverdin even in the control, the biliverdin reductase wild type mice. So I also think that maybe there is a need to revisit that concept that biliverdin is so water soluble that it's eliminated so fast that it can't be accumulating. Maybe that's not always actually the case.
Because in a way, your data are telling us that maybe bilirubin is more powerful than biliverdin, because even if we have a little bit of an accumulation of biliverdin, we may not have as much protection as when we have bilirubin, because this is what you show in your experiment.
That's right. Yeah.
But perhaps the fact that
and also in the chemical tests, when we tested that, I'd go back to the very beginning, when we tested the chemical antioxidant activity of the pigment. I mean, there's no doubt that biliverdin is not as strong an antioxidant on a mole-per-mole basis compared with bilirubin. At least for radical oxidants. 2-electron oxidants might be a different scenario, but certainly not for radical oxidants.
Yes. Just another question in relation to these experiments. So you see that, or it looks like the presence of bilirubin in the unstable plaque is important. I mean, we need bilirubin to prevent the plaque to become unstable, correct?
But you're actually opening a can of worms here, Roberta. So, and we need to go back to heme oxygenase 1. So, as you know, one of the strong inhibitors of heme oxygenase 1 is heme itself. Well, one of the characteristics of unstable plaque is intraplaque hemorrhage. So we get bleeds in those plaques. And of course, with that, we get the presence of red cells. And we have infiltrating inflammatory cells, including macrophages. So we get an induction of heme oxygenase 1 in unstable plaque. And in fact, there has been a very influential publication that suggested that in this scenario, atherosclerosis, heme oxygenase 1 is actually detrimental, because they could show that the extent of heme oxygenase 1 induction correlated with plaque instability in humans. What I think is that what they reported was nothing but a consequence of intraplaque hemorrhage. And it is well known that increased intraplaque hemorrhage is associated with increased plaque destabilization. So even though there is literature to suggest that induction of heme oxygenase 1 and its resultant degradation of heme to bilirubin may be detrimental, we argue the opposite way. And we say, in fact, that is part of the defenses to the presence of intraplaque hemorrhage, during which a damaging molecule, heme, is converted into a protective molecule, namely bilirubin. And hence, yes, the presence of bilirubin in unstable plaque is important from a plaque stabilization process, and that's likely through an anti-inflammatory, antioxidant mode of action.
Before I move to the next question, you stimulated another one, and it's something that we were discussing offline, the substrate for heme oxygenase activity. In your experience, do you think that the heme that is used by heme oxygenase to produce the bile pigments only derives from hemolysis, or do you think there is also an intracellular source of heme that is used in stress situations when HO1 is activated?
So, look, 80% of our body's heme is present in hemoglobin. So logically, you would expect that red cells hemolysis is a major source of substrate for heme oxygenase. It has to be. Is it the only source? I think in all likelihood, no. And in fact, we published many years ago cellular studies where we looked at long-term effects of elevated heme oxygenase 1 expression in a cell system. What we observed is that also heme-containing proteins like cytokines become decreased over time. So clearly, if you have heme oxygenase activity in a biological systems, it will essentially, it will use heme from all sorts of heme proteins. Again, quantitatively, I'm sure that hemoglobin is always going to be a major player, but this does not mean that other heme-containing proteins that, where the heme has to turn over, do not provide a realistic source for heme to be converted into bilirubin.
Yes. So, yeah, I'm still thinking about your description of the atherosclerotic plaque, because all we think about is the accumulation of the fatty tissue and perhaps the inflammatory cells, but we never think about the hemolysis that is also occurring locally. So this is, I think, an important point that maybe joins all the different elements and all the different processes that are occurring there. Also because the release of heme will be, again, a stimulation for free radicals production, and therefore, again, the usual process of biliverdin and bilirubin being able to protect there.
Yeah. And if you look at the human disease and you do histological analysis of, let's say, a carotid lesion using endarterectomy specimen, it is very common. So you do an iron stain. So it's very common to see iron deposits in all the lesions that have healed, and you can see several layers of, it's almost like, a history of the disease that vessel has undergone. And there's no doubt that, iron deposits are very, very common, certainly in the human disease, arguably much less common in animal models of atherosclerosis. So, but I do think in the human disease, there's undoubted evidence for the presence of heme and hence substrate for heme oxygenase.
So we go back again to your original article where you were studying at the same time bilirubin synergizing with vitamin E to protect against the radicals and oxidative stress in the cell membrane. So as you know, I'm a nutritionist, and I find this finding fascinating for different reasons. The first is that vitamin E is a fundamental antioxidant that we have to introduce with diet. For example, you find it in olive oil, in nuts, in seeds, in wheat germ. And this is in contrast with the bilirubin, which is actually produced by our body. It's an antioxidant that is produced by our body. And the third fact that to me is very interesting is that an endogenous antioxidant and an exogenous antioxidant, such as vitamin E, can work together to protect the cell membrane against damage. Cell membranes or perhaps other cellular structures. So what is in general your view of this concept? And of course, there are consequences if we think about this, because we may have situations where bilirubin production is decreased for some reasons, or we have a lack of vitamin E in our diet, and therefore the fact that the two of them work together, but maybe there are situations where one of them is going to be decreased. What do you think could be the consequences, and what do you think about this concept of synergism?
Yeah, it's an interesting issue, isn't it? And yes, indeed, bilirubin and vitamin E or alpha tocopherol can work cooperatively to prevent oxidative damage to lipids. So we've demonstrated that very, very clearly. But you need to keep in mind that bilirubin does not require vitamin E to protect lipids from oxidative damage. And in fact, I'm not aware of literature that would suggest that low levels of vitamin E decrease the antioxidant activity of bilirubin in vivo. I'm not aware of that type of articles. When we look at the converse situation, it gets a little bit more complicated, because there are indeed situations where vitamin E requires bilirubin or other antioxidants, and we term those antioxidants co-antioxidants. It requires it to effectively prevent lipid oxidation. That's predominantly shown in lipoproteins. And the larger the lipoproteins, the more pronounced this requirement of vitamin E for a co-antioxidant is. So still observable in HDL, small particles, clearly observable in LDL, and very observable in VLDL or chylomicrons. Does it extend to membranes? We don't think so, because they're too small for this interaction to be required. So in the context of atherosclerosis, we were interested in that because, as I mentioned earlier, lipoprotein oxidation, according to the oxidation theory, was thought to be an initiator of atherosclerosis. And in that sense, we did think that the synergism between bilirubin and vitamin E could be really important. And it could still, in part, explain the epidemiological studies showing an association between high bilirubin, high circulating levels of bilirubin, and cardiovascular disease events. So if we then talk about vitamin E, and I'm not a nutritionist and I've gotten into very hot waters over this with my views. So the importance of dietary vitamin E, I believe, should also be looked at least in part, from a health benefit point of view, with the association of vitamin E, consumption of vitamin E-rich foods, because as you pointed out, those are normally healthy foods. And I think that interpretation is in fact consistent with the overwhelming evidence that shows that low vitamin E levels and diets low in vitamin E are associated with increased vascular disease. Yet, when you then try and go in the other direction, you come in with vitamin E supplements, it is now very clearly established that do not decrease cardiovascular disease. In fact, if anything, vitamin E supplements increase mortality, it does not decrease mortality. So, yes, there is cooperation. I see it much more from a bilirubin perspective than from a vitamin E perspective. And certainly, bilirubin's lipid antioxidant activity can be enhanced in the presence of vitamin E. So in the case of large proteins, not necessarily membrane cell membranes. Yeah, but I don't necessarily think that applies in the other direction.
Very interesting. Yes. So I have a final question. There is a recent work being published in the literature and also work from a Japanese company. They are working on new therapeutic molecules that are based on bilirubin conjugates. And they are studying these new molecules in the context of cancer, trying to reduce cancer and tumors, and also in inflammatory conditions. So the fact that they are bilirubin conjugates hints at the fact that perhaps bilirubin by itself is difficult to handle. What is your view about these recent developments about bilirubin as a potential therapeutic molecule for the future, or bilirubin-based therapeutics?
Yeah. Look, I mean, they raise very important and interesting issues. The research that you referred to, I think, was actually a Korean lab, and Japanese are catching up on that as well. They have generated these so-called bilirubin conjugates, and they've used different conjugates, PEG and hyaluronic acid, for example. Interesting thing, and maybe I'm just, pointing out the detail, it's actually not really a bilirubin conjugate that they use. If you look at the underlying chemistry, they actually modify the bilirubin. First, they convert one of the carboxylic acids into an amide, and then they form a conjugate. So they're not, in fact, true bilirubin conjugates, the kind of bilirubin derivative conjugates. So the concept is that they add a very polar moiety, like polyethylene glycol or hyaluronic acid, to this water-insoluble molecule bilirubin. And then that leads to a self-assembly whereby the hydrophobic water-insoluble part, the bilirubin, is in the center of my cells with all the polar residues sticking out, right? And indeed, when you use these conjugates, they are readily water-soluble, and they, automatically form these micelles, and these, the groups they characterize these micelles. So just before we talked about, how does bilirubin act as an antioxidant in vivo? And I said it is actually able to reach that interface between a protein-bound form and a lipid form, and a question whether hydration or a hydrated bilirubin molecule is needed hypothesis. I would have thought that putting the reactive molecule into the center of a micelle will not facilitate its interaction with a lipid-bound vitamin E molecule, for example. It should be doing the opposite. In fact, the very polar structure around the micelle will almost ensure that complex does not get even close to a lipid environment. Will it incorporate into proteins? Again, I doubt it, because the micelles are, a structure by themselves. So I think fundamentally, to me, and, I have a very biased view about this, but I think that approach really changes conceptually the location and likely interaction of bilirubin with biological structures. When you look at the papers, at least as far or as little as I have done, I haven't done a comprehensive literature review of that. The characterization of that conjugated bilirubin with regards to antioxidant activity is very rudimentary. They use non-specific methods, and they only do test tube experiments that are of unknown physiological relevance. And I've not seen, maybe I've missed some studies, but I've not seen anything biological, an antioxidant test, for example, in a biologically relevant fluid like plasma or something like that. So I think the concept, of making a difficult to work with water-insoluble molecule water-soluble is good. What is also good about this approach is that, and maybe that's really where the advantage will be with that approach, is that they can provide additional cargo to those particles. For example, that targets them specifically to a particular cell, like a cancer cell, and hence provide some targeted activity rather than, general activity. But I'm not convinced that this is the approach to be used to, provide bilirubin in an antioxidant fashion comparable to the bilirubin that is in your body and in my body. I also know that, these things need to be administered intravenously. That's not a great way to go about it. So I do think probably there are alternative approaches to consider in terms of formulating bilirubin. And to me, an obvious approach to pursue would be the combination of bilirubin with bile salts, because that's the situation we have in bile, where we know that the concentration of unconjugated bilirubin is several times higher than it is, in a PBS buffer or a pH 7.4. And we know that we can achieve concentrations in, up to 30, 50 micromolar of unconjugated bilirubin in the presence of bile salts. And so to me, that type of formulation seems to me a more obvious way to go, particularly because bile salts are being used for other difficult to use, compounds in therapy and pharmaceutical industry.
And because they are produced in our body. So
and yes, that's correct. Yeah. Anyway, so that some are thoughts. Certainly an interesting area to look at, but I think it does deserve some more careful analysis of what they think the mode of action of bilirubin in these complexes indeed is.
So you're not convinced that bilirubin in this, or a bilirubin derivative in these conjugate molecules can still maintain the antioxidant activity and perhaps...
So No, I mean, it can to some extent, but from the studies, and again, I should say the limited number of papers that I've read on this, I'm not convinced that this activity is likely to be relevant under a biologically, relevant condition. So they've used test tube conditions that I consider to be not very physiologically relevant.
Yes.
And when they do comparison with other antioxidants, including bilirubin, the way I've read, the way this is done is just inappropriate. They don't compare apples with apples. So we don't actually know, even from where people have reported antioxidant activities of these conjugates and the nanoparticles, we don't really know how that compares with anything else, including bilirubin, unconjugated bilirubin.
Yes, very clear. I think we are ending our very interesting conversation. I've learned an enormous amount of information. I'm so glad you accepted to be with us today. And I really thought I knew a little bit about bilirubin, but definitely I realized that I need to go back and look very carefully at all the studies that you published and all the insights that you produced over the years. So I thank you ever so much for being with us. You raised some other points that are very interesting. You were talking about myeloperoxidase, for example. I'm very interested in that subject. And if you are available, I may come back and ask you to participate in another podcast. If I can take you out for a few hours from your retirement and your enjoyment of retirement. Thank you so much again. I hope that, you are, getting on with your studies. I see that you are still very interested and very implicated in science, although you may be working from home. And I thank you again for your participation. And I hope that our podcast will help everybody who is interested in bilirubin to learn more about this subject.
Pleasure has been all on my side. Thank you so much, Roberta.